Welcome to Faces of IICD. Today I am interviewing Beatrice Grauman. Can you introduce yourself with your name, your role, and the lab that you're part of at IICD?
Yeah, my name is Beatrice Grauman, and I am a PhD student based out of the medical campus here at Columbia in the integrated program for cellular, molecular, and biomedical science, and I am in the Vicković Lab at the York Genome Center. Yeah, so my background is kind of unconventional. So I grew up in a small town on an island 2 hours off the coast of Washington State. Where I knew I was interested in medicine and health and that type of science. Volunteering at the University of Washington Freddie Harper Labs, where I worked with a fantastic postdoc and studied larval sea urchins. That got me really interested in this concept of like how cells can grow at different rates and how things can perturb normal behavior. And so because I wanted to go into the sort of human side of research, I kept thinking back to kind of like what is the most applicable thing. And in my high school mind, the first thing I thought of when I thought about dysregulated growth was cancer. When I got to Columbia, the thing that really stood out to me here and with Sonia's lab is they do this multimodal approach to studying cancer. So we not only are doing the traditional sort of molecular biology approaches, but we're also using spatial transcriptomics, which is what I work on, in order to sort of interrogate not only like what's happening, but like what's happening in the context, right? What I am doing is a combination of technology development and also disease research. And it is in the context of colorectal cancer, which is one of the most deadly forms of cancer. One of the things that we want to understand is how do we characterize this disease and tailor treatments, and also understand how does that develop even within a category, right? I want to understand these cell state transitions within the sort of larger category of like, this is this cell type. And so in order to do that, I'm using this technique called spatial transcriptomics and also trying to improve on that technique. So at the same time as I'm trying to characterize the disease and its development, I'm also trying to figure out what is the best way that we can characterize this disease. So I think the thing that gets me the most excited is being at this intersection of technology development and cancer research. So for a long time, I thought that I might want to go to medical school or do an MD PhD. And I thought I was sort of confusing my interest in medicine with the desire to practice medicine. What I really like about what I'm doing now and what's so exciting about it is that not only am I answering questions that, using new technologies that haven't been answered before, but then I'm tailoring the technology to the question. And I have the resources and the mentorship that can help me do that, so I can answer questions that have never been able to be answered before. And that's a really exciting place to be in. So some of the best advice that I got when I was younger is don't just find out what interests you, find out what doesn't interest you. Yeah. In college, I was studying cancer and therapy resistance. After college, I decided to apply for a technician position at Rockefeller University, studying something completely different. Because I was like, I love cancer research, I think it's awesome, but do I know that I want to commit to that forever? I haven't tried a lot of other things. So I went to Rockefeller and studied viral infection for a year and a half. And what I figured out is that it's cool, but I want to go back to cancer research. And so when it came time to apply to grad school, I really felt confident in this is what I want to do. And so, yeah. Follow your curiosity and figure out what you don't like as much as you do like. My fun fact is that my wife and I are both at Columbia, and she's an MD PhD student, and we met doing cancer research at MIT. Yeah, we all came together at Columbia.
That's amazing. Well, thank you so much for interviewing with me today, and we're so happy to have you. And yeah,
Awesome.
Thanks everyone. We'll see you next time.